Eve, can you hear me? Vutu, can you hear me? Yes, it's a struggle. So where are the other class members? They're lost. Jenny, I can see you. Hi. This is for metabolism, but whoever want to join metabolism, but whoever want to join can join. Jenny, if you are free, you can still join. It will help you in semester 3. Everyone who want to join, who are in biomolecules, yes, you found yourself here because you are among the subscribers and you can see me live. So that's why you found yourself here. But nothing is lost. If you can be patient, you can just stay, Jenny. You can just stay. And so work Simon talking about they can't hear me. So it's okay. You can just listen. Vutu, can you share with others? I know it's a challenge, I'm not Gen Z. I have to admit that, things relating to these computers is bringing a bit of complication. But it's okay. So, Jenny, yes, we welcome you. As we wait for the others, it will be helpful in semester 3. I've shared with them they are coming. Thank you, Evelyn. Thank you. I thank you so much. I thank you so much for the subscriptions, for the viewing. And I hope it will be helpful to the students. So, as we wait for the others to come in, I appreciate all the efforts the students are making. I appreciate. So let's wait. Yes, I can see. Hello, Dr. Sandeep. We are here. We are going to discuss about metabolism, metabolic pathways. Whoever want to listen, you are allowed. Nobody is limited to listen, So if you have time, we just listen as we are going to revise the metabolic pathways or the metabolism of those biomolecules we've been discussing in the channel at the introduction part. Good. Dr. Sandeep, I can see you. Thank you so much. I'm fine. So even if you are doing Biochem 1 or Biochem 2 and you found yourself here, you are welcome. We are welcome. So let us just learn together. Nobody is limited. The contents will be helpful. Even for the students who haven't done metabolism, it will still be helpful. So nobody is limited. So let us just wait for others to come. Thank you, Jenny. Thank you so much. Just let anyone join if they want to join, even the medicine students. I know it will be helpful in semester 3. As we wait, Vutu, kindly tell them to come. Dr. Sandeep, we are here online for revision with the students for metabolism. If it will be helpful to you, it's also okay. We'll appreciate. Yes, Ruth, I can see you. Ruth Midega. Welcome. Let's come on board, please. We have only 16 minutes to 10. So, Vutu, kindly just tell them to rush. It's just a matter of clicking a link, please, so that we don't waste much of the time, because it's even late hours and we might take longer when we lengthen even in revising and understanding the questions, which is fine. because this is the last chance we have, because the exams are starting next week. So it's very important they join. So let them come, Vutu, as you just keep on telling them to come. Yes, yes, Ruth, welcome. Please tell your friends. Oh, Dr. Sandeep, welcome. Where are you from? I'm from Kenya. My country is Kenya. Where are you from, Dr. Sandeep? Thank you so much for joining. Just tell us the country where you are watching from. We are appreciating. Dr. Sandeep? Ah, perfect, perfect, perfect. Even the Indians are represented. Thank you so much. Thank you so much. From India, we are saying hi. We appreciate all the efforts of viewing, commenting. We are together in this. Biochemistry, we want the students to understand. Oh, perfect. Somebody from Portugal, welcome, welcome so much. We welcome you all. We welcome you all. Thank you so much. Watching from Portugal, watching from India, we appreciate. Please, we appreciate. Hi, yes, Bosley, you are from? Ah, welcome. Welcome. We welcome you. We are from Kenya. Oh, wow. You are from US. Vutu, you are from US. Okay. We welcome all. Everyone is watching. We appreciate. We appreciate. Somebody from Palestine, somebody from India. We welcome you all. We welcome you all. Together we are going to learn. We try to simplify biochemistry because most students have seen it as a difficult course. So the main objective, no, we can't start the topic before the students come. It's a revision question. Oh, somebody from Haiti, welcome. It's a revision session for the metabolic pathways, for metabolism, for the biochemistry 2 students. So the possible questions, how they need to answer so that they don't miss a point at the time they're doing exams. Oh, wow. I thank you so much, Dr. SK Sainape Juma. Hi, welcome. Oh, the students are here, but they are still at 21 and they are always 40. So I don't know. Oh, from Eritrea, we thank you so much. We thank you. Bosle, we are soon starting. We are soon starting. So I told the students, yes, we'll pray for Palestine. God is with you. We are from Kenya. We appreciate. Jaipal, I'm Kenyan. I'm Kenyan. Dr. Sandeep, you want to ask a question? So we appreciate. Vutu, can you tell me when most of the students have logged in so that we can start by revising the possible questions? So I told you to have a paper and a pen, or a book and a pen. A paper won't be enough because we are going to explain any of those questions which you might see in carbohydrate metabolism, in lipid metabolism, in protein metabolism, and then we'll be able to see how we need to answer them. So for the medicine students, we did biomolecules last week, revision questions, but still you can join so that we can also try to understand much about the metabolic pathways. So we just want to have the revision questions. Yes, you are a B Farm student. Ah, from India. Indians are many. Thank you so much. Dr. Sandeep is asking, what are your questions? What we are trying to do here is we are trying to revise the possible questions, based on the basics of the metabolic pathways. So if we talk much about the carbohydrate metabolism, we are going to start much on the glycolytic pathway, a bit of the questions how the students need to answer, the gluconeogenesis, the glycogen metabolism, the TCA cycle, the pentose phosphate pathway, and how important those pathways are. And then we can move to amino acid metabolism, the degradation pathways, the glucogenic, the ketogenic amino acids, and the diseases, the metabolic disorders related to any of the defective enzymes on those pathways. And then we are going also to move to fatty acid, oxidation, and synthesis, and then the formation of ketone bodies. So a bit of what the students touch the students in class in relation to metabolic pathways, because we already started off with the introduction to the biochemistry, where we were looking at the biomolecules. And we mentioned that in the biomolecules, as much as they are the polymers, so we dwelt much on the monosaccharides in the basics of the carbohydrates. We started with the monosaccharides, but then we went to the disaccharides, the polysaccharides. But in those biomolecules, there are organelles. And these organelles, the metabolic pathways take place there. Because we started, if you look at my slides, my sessions, we did introduction for this channel by mentioning how important and defining what biochemistry is, chemistry of life. So we had to start, for you to have life, there has to be those organelles. So we talk about the cell, the basic unit of life. And what involved the cell? The organelles, the subcellular components. And those subcellular components, we said, those organelles, a metabolic reaction takes place there. And that is what makes us live. So in those metabolic pathways, whatever we are going to discuss today, the enzymes are involved. And the enzymes are the ones which catalyze those pathways. So if those enzymes, which are protein in nature, get defective in any of those parts, then it leads to those metabolic disorders. So for this session, we are dwelling much on the metabolic pathways. Much of the introduction I've done on my slides. So in relation to my sessions and what we cover in class, the bit which we are doing in the channel is that bit which the students get hard to understand. So we are not bringing everything to the channel, but that bit which makes it difficult for the student to understand. Because for the 10 years plus which I've been teaching, I've been having challenges of the students who do not understand what they need to understand, and then some even drop off because they already feel that it is difficult. But it has to start from the simplest part, the biomolecules, drawing a simple structure of a L and a D as a linear or Fisher's projection, moving to the Howarth, the cyclic form, because the disaccharides, the Howarth, will always promote solubility. It will increase their solubility. So we've started from the biomolecules, if you look at my previous session. So Dr. Sandeep, today, I'm just going to deal with metabolism. And the possible questions I'm just going to read. For example, if a student finds such a question in exam, how do they need to attend to that? AC80 Army. We are not talking about beauty here. We are seriously studying, eh? Thank you so much for the compliment. But we are seriously studying, eh? I appreciate the compliment. I have full hair on my head. I have full hair on my head. That is only a dreadlocks. You see? I have full hair. So, Vutu, how many have come? Please? Jenny, you are laughing. Yes, maybe she is seeing. They are seeing. That is my hair. But I'm not here to discuss my hair. Thank you so much for saying I'm beautiful. But I have hair. Hello. Yes. Shabazz Khan. Vutu, can you tell me how many have come on board, please? I need to start with the question. Hello. Shabazz. So I've just tried to mention much what we are going to do for this, why this session is here. Oh, thank you. Thank you, the Indians. You are well represented. I appreciate. I appreciate so much. I appreciate. So thank you so much. Ah, Vutu, good. Frederick, I can see you. Gashanja, hello. Tell others we are starting. Do you have a pen and a paper? So I didn't separate them into the carbohydrates, the lipid, the proteins, but generally, I'm just going to mention a question and I tell you what I expect of you. So what we need to write, if you see such a question, how you need to put your points. You know, the metabolic pathways, just by being mentioned, using structures describe a certain metabolic pathway, no stories. Just go straight to the structure and write the correct enzymes. That is what I always say. Because of specificity of enzymes, you can't give a reaction which is catalyzed by isomerases to a transferase. That one doesn't work. So it has to be exact. Specificity of enzymes, we did in Biochemistry 1. Substrate specificity, optical specificity, reaction specificity, group specificity. It was preparing us for Biochem 2, the metabolic pathways. It is good to mention the correct enzyme. Very important. So we are going to start. These are the questions I won't answer. I'm sorry. We have to start. I think 2 more have, bundles also a problem. So Vutu, we are going to start straight away. I told you last time I'm going to read them from my laptop because I'm not good in these IT things. So I don't know how to upload. Last time the student told me, Madam, you can't upload anything from YouTube. So let us start by looking at the first one, because the pathway which is always very commonly asked about is always, you have to know glycolytic pathway, of course. Before you know any other pathway, glycolytic pathway you have to know. So I'm going to start with the short answered questions. And in the short answered questions, I'm going to explain in depth if any of those possible questions are asked based on the pathway. So the glycolytic pathway, the first question which comes is, using structures, show the ATP utilization steps in glycolysis. Using structures. I told you none of my question I'll ask without using structures. Only if you need to discuss any of the metabolic diseases related to a pathway. But most of them using structures, correct ones, because structures we learn in Biochem 1 to help us understand whatever happens in metabolic pathways. So using structures show only the ATP utilization steps in glycolysis. Just by generally defining what glycolysis is, as we did in class, you are breaking down D-glucose to pyruvate with the main objective of generating energy in form of ATP. But you also have to remember the glycolytic pathway having the 10 steps. We discussed that it involves the 3 stages. The stage number 1 was to destabilize our glucose. Remember, it is D-glucose, optical specificity of enzymes. We discussed that one in Biochemistry 1. How specific enzymes are. In carbohydrate metabolism, the enzymes will work on the D sugars. In amino acid degradation or protein metabolism, they work specifically for the L amino acid. So in glycolytic pathway, we are breaking down D-glucose to pyruvate. So stage 1, we mentioned, it has to be destabilized. And that is why stage 1 had step 1 to step 3. The step 1, we had our D-glucose itself, converted to glucose 6-phosphate. And we said the hexokinase has high affinity for glucose than the glucokinase. Because if you look at the substrate specificity, we talked about the fructokinase, the galactokinase, but then in this case, the hexokinase is going to catalyze that step. We are talking about stage 1. We are destabilizing our glucose. So I'll have to explain the glycolytic pathway first, and then I'll mention whatever happens, what we need to answer in this question if it's asked. So stage 1, the main objective of stage 1 was to destabilize our D-glucose, such that at the end of stage 1, that is in step 3, we have fructose 1,6-bisphosphate. But still, in the glycolytic pathway we mentioned, it involves several reactions. There are phosphoryl transfers, there are phosphoryl shifts, there are isomerization, there are aldol cleavages, which we need to discuss. So at stage 1, we have the hexokinase. At step 1, the irreversible step. Remember, it was an ATP utilization step. It comes in as adenosine triphosphate, then goes out as adenosine diphosphate because one phosphate has been used to phosphorylate our D-glucose at carbon atom number 6, and we have glucose 6-phosphate. Then step number 2 is reversible. We have phosphoglucoisomerase. We talked about functional isomers in Biochemistry 1. They're of the same molecular formula, C6H12O6, but their difference is based much on the difference in functional group. So if you look at glucose 6-phosphate, glucose is already an aldose, specifically an aldohexose. But then it is converted to fructose 6-phosphate, which is a ketohexose. So the enzyme, based on the reaction, because the International Union of Biochemistry classified enzymes in those 6 classes, that is an isomerase. It is converting a ketose to an aldose to a ketose. So the second step is reversible, and it involves an isomerization, so the phosphoglucoisomerase. Then you have the third step, ATP utilization, still a kinase. Yes, the phosphofructokinase will catalyze that step. ATP utilization step is there. So that is stage 1. We end up having fructose 1,6-bisphosphate. But remember the kinases, those enzymes work well in the presence of magnesium ion and manganese ion. So what it tells here is already the question is answered, because it talks about using structures. You have to show only the ATP utilization steps in glycolysis. So it involves the stage 1 reaction, except the step 2. So step 1 and step 3 is what you show here. And here I'm giving my small marks, like half-half. Because if you see, I say only, and it is 5 marks. Putting the ATP well, the arrow has to show well. Going out, ADP, because one phosphate has been used to phosphorylate our glucose at carbon atom number 6, and the other one has been used to phosphorylate our fructose 6 to carbon atom number 1 to form fructose 1,6-bisphosphate. So the ATP utilization, consuming something which goes in, the ATP utilization steps, step 1 and step 3, which is catalyzed by hexokinase and the PFK. In this case, don't give me the isomerase reaction, the one for phosphoglucoisomerase, don't give me that. I only need step 1 and step 3. Be precise to the point. Draw correct structures. Write the correct enzyme. Imagine if you give me a wrong enzyme, the whole process is wrong. Because I just said, based on how the enzymes were classified, and based on the reaction they catalyze. So you can't just guess a different class of enzymes catalyzing a wrong reaction and you get an answer. It won't work. It doesn't work like that. So you'll also proceed with stage 2. And we saw in stage 2, it involves the aldol cleavage, and there is also an isomerization taking place there. The aldolase. In stage 2, the main objective was to generate the 2 trioses, the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. So the interconversion of the ketotriose and the aldotriose is an isomerization. So since both of them are trioses and both of them are phosphorylated, the enzyme is triose phosphate isomerase. And at step 4, we have the aldolase. So that is how important the stage 2 is. We have to generate We have to generate the 2 trioses. Then the stage 3 will accomplish the main objective of glycolytic pathway. We'll be able to generate energy in form of ATP. So in this case, we are talking about a process where our D-glucose has to be broken down to pyruvate with the main objective of generating energy. So this question says, kindly using structures, describe the ATP utilization step. Go straight and draw the structures. After drawing the structures, write the correct enzyme. Magnesium ion or manganese, very important for the kinases to work. So the arrow should clearly go in as ATP, out as ADP. Still, on glycolytic pathway, a question can come, just show only the phosphoryl shift. You know, the phosphoryl shift, the mutases will do the work. So where we have the phosphoglycerate mutase shifting from the 3rd carbon to the 2nd carbon, you have the 3-phosphoglycerate, and now the other one is 2-phosphoglycerate. There can be phosphoryl transfer, whatever the kinases are doing, those are phosphoryl transfers. A question can come in different way. Just show the isomerization steps, and you can see the triose phosphate isomerase together with the phosphoglucoisomerase. And then sometimes the question can come, only show the ATP generation steps, or only show the substrate level phosphorylation steps. We are going to see possible questions which might come. But for this one, whatever I need was where the hexokinase is catalyzing the step and where the phosphofructokinase is catalyzing the step. So I said I didn't separate the carbohydrate from the protein from the lipid. We are just going to ask those questions in random. The second point says, mention where beta oxidation takes place. We are still on short answered. And list the steps involved in beta oxidation of fatty acids and the enzymes involved. So you know very well what happens during the beta oxidation. Beta oxidation of fatty acid is the degradation of fatty acid, in other words. So when you talk about the beta oxidation, whatever we discussed was, before beta oxidation takes place, those fatty acids have to be activated. And by activation, what we mean, we were adding a co-SH group. The thiokinases were involved there. We were adding a co-SH group. That was the first step. That question I asked in the cut, you know, the cut was only 10 marks, the second cut. And it was only beta oxidation, the 4 steps is what I wanted. So the bit which we talked about, the activation, and the bit which we talked about the carnitine transferring to the inner mitochondrial membrane, I didn't mark. I was starting at the point where the enzyme acyl-CoA dehydrogenase is involved. The FAD utilization step, the first step. So in this case, it says mention where beta oxidation takes place in the inner mitochondrial membrane. And you list the steps. Just by listing and the enzymes. The first steps is an oxidation step. It involves a dehydrogenase. The second step involves hydration. The third step, the second oxidation. But now, NAD plus dependent, NADH is generated there. And then the last step is thiolysis. So if you mention the enzyme and the steps, so for the enzymes and the steps, I give half a mark because it's 5 marks. So if you mention where it takes place, you get 1 mark, but then the steps and the enzyme, you get half. So it's like 1 mark. If you mention the step, half, the enzyme half, 1 mark. So just go straight to the point. Don't write stories. Just put whatever you have mentioned here. Here there is no structures. I've said clearly. Mention where beta oxidation takes place and list the steps, because that one has to come into short answered. It can't come in the long answer. You know, if you come to the essay, then definitely you need the structures, and then at last you need the correct enzyme. So it will take possibly 10 marks. So in this case, it falls under short answered. You just go straight and mention where beta oxidation takes place, the steps by mentioning the oxidation, the first oxidation, the enzyme itself, FAD dependent. And it has to be the FAD. Generation of FADH2, it has to be in step 1. Don't try to change it your own by bringing NADH. No, it won't work like that. FAD, generating FADH2, which is used as a reducing equivalence for the electron carriers. And then the second step, hydration step. The third step, another oxidation, NADH generated there, another electron carrier. And then lastly, thiolysis. We are cutting our even-chained fatty acid, which is saturated. And that is why we learned first of the biomolecules, because we saw the structures of the saturated and unsaturated fatty acid. So at that point, we are degrading, we are catabolizing an even-chained fatty acid, which is saturated, of which the even-chain fatty acid, after degradation, will give us acetyl-CoA. For the odd chain, remember, it will give us propionyl-CoA together with acetyl-CoA. The acetyl-CoA can go to the TCA cycle, but the odd chain fatty acid, the propionyl-CoA, will be converted to succinyl-CoA, which will at last also go to the TCA cycle. So just be precise to the point. Question number 2 says, using structures, show the steps involved in fructose entry into glycolysis. We said at the point you are digesting sucrose, that is the small intestine, the sucrase, it is going to generate glucose and fructose, because those are the components of sucrose. But then the glucose will go through absorption, because I said it starts with digestion. Absorption is when oxidation takes place. So the glucose will go straight to glycolytic pathway. But the fructose, absorption itself is a bit slow because of the keto group at the second carbon atom. So this fructose, it can't go straight to glycolytic pathway, but it has to be converted to one of the intermediates of the glycolytic pathway. And that is why we are saying our D-fructose, the enzyme being fructokinase, is going to convert our fructose to fructose 1-phosphate. After conversion to fructose 1-phosphate, fructose 1-phosphate is not an intermediate of the glycolytic pathway. So the other option can be a mutase. The mutase will shift the phosphate to carbon atom number 6, and you'll form fructose 6-phosphate. It will enter glycolytic pathway, because at stage 1, the fructose 6 will be converted to fructose 1,6-bisphosphate, and that's how it will go and generate energy in form of ATP. The other option is, apart from converting our fructose 1-phosphate to fructose 6-phosphate by mutases, the fructose 1-phosphate is broken down into the trioses, aldolase doing the work. The upper part of fructose 1-phosphate is dihydroxyacetone phosphate. The lower part is glyceraldehyde. The kinase will do the work. And then it will add a phosphate to the glyceraldehyde. It will form glyceraldehyde 3-phosphate. So that glyceraldehyde 3-phosphate can now connect to the glycolytic pathway. So you have other options as long as you put the correct enzyme, and you'll be able to understand how also, as you consume the sucrose, which is the common sugar which we use every morning, which is a disaccharide, you have to learn it in the biomolecules, the components which I say you have to specify the alpha D glucose and beta D fructose. At the point of small intestine, the sucrase doing its work, yes, D glucose will go straight to the glycolytic pathway, but the other one, it has to find an intermediate. So that is how fructose provide energy. Point number 3 says, using structures show how asparagine connects to TCA cycle. We mentioned very well that the TCA cycle, the main objective is to generate the reducing equivalences. The NADH and the FADH2s are used as electron carriers in electron transport chain. So if you look at those amino acids, the asparagine, aspartate, asparagine is the amide of aspartate. Still, if you want to connect this one, you can still talk much about how the amino acids act as non-carbohydrate precursors in gluconeogenesis. At the fasting state, the body is on starvation, so it needs to find glucose. So the asparagine, the aspartates will help much, because the asparagine will be broken down to aspartate via the asparaginase to release the amine group which was present on the R side chain. But then it will be further converted to oxaloacetate, which is an intermediate in gluconeogenesis and an intermediate in the TCA cycle. So it will enter the TCA cycle through the formation of oxaloacetate. That is how it connects. And I said, in bioinformatics, we talk about system biology. The interactions between metabolomics, genomics, transcriptomics, proteomics, all these metabolic pathways, they are interconnected at one point. And we saw the amphibolic role of glycolytic pathway. It was able to provide materials for other pathways. That's how important these metabolic pathways are. And that is what makes us alive, or to have life. Because those metabolic pathways, those organelles, the enzymes have to do their work. So the connection of asparagine to the TCA cycle is when the asparagine, where the asparaginase will convert it to aspartate, and then aspartate to oxaloacetate. Oxaloacetate will connect to the TCA cycle. At the point the acetyl-CoA comes in from the glycolytic pathway, remember, in aerobic state, the pyruvate is converted to acetyl-CoA. Via the pyruvate complex of enzymes, 3 of them, it will be converted to acetyl-CoA, which has to enter and combine with oxaloacetate to form citrate. So at that point, the asparagine will be broken down to aspartate to oxaloacetate, and then it will connect to the TCA cycle. So that's why we talk about asparagine being glucogenic amino acid, because the degradation product is an intermediate of the TCA cycle. State the roles of the following enzymes. It just try to remind you. So if you're told to state the roles, don't write stories. Just say this enzyme convert this to this. The transaldolases, you have to remember, not exactly should I give the correct answer, the same answer, not the correct answer, the original answer, but you have to remember in the non-oxidative phase of the pentose phosphate pathway, the transaldolases, the transfer of the number of carbon atoms, how many the transketolase does, whether 2 or 3, you have to go back and read. But then they work well in the non-oxidative phase, where we generate the ATP, where now the pentose phosphate pathway acts as a shunt, where now we generate the ribose from the ribose 5. So in this case, the transaldolase just mention the transfer of the number of carbons from an aldose acceptor to, from a proton, from a keto donor to an aldose acceptor. So just by explaining that, some students will always explain whether it converts a certain sugar or a certain monosaccharide to another one, which is also fine as long as you do the correct one. Pyruvate carboxykinase, the main objective of asking these enzymes, some students don't even know where the enzyme falls, which of the metabolic pathways, which step does it catalyze, what product is produced from that step. If the enzyme is defective, which type of a metabolic disorder will it lead to? So it's very important to know when such questions are asked, which pathway, can you possibly tell which of the pathways is being mentioned here? So the pyruvate carboxykinase, if you remember the gluconeogenesis, the bypass steps, the bypass one has two enzymes. There is an oxaloacetate in between. The pyruvate and oxaloacetate, from oxaloacetate to PEP. So the first one, since the oxaloacetate is 4 carbon atoms and pyruvate is 3 carbon atoms, it's a carboxylase. But then the carboxykinase comes between oxaloacetate and phosphoenol pyruvate. So you just mention the conversion of oxaloacetate to PEP. Don't write a lot of stories, 1 mark. Only if you are asked much in essay, but there I'll mention using structures. And you have to tell me all about the GTP and GDP, the presence of biotin where it is needed. So we have to be specific with these enzymes. So the carboxykinase is found in the gluconeogenic pathway. You need not to write that, but you need to tell me it converts this to this. The alpha ketoglutarate dehydrogenase, you saw very well at what point it is in the TCA cycle, the second step, where the NADH is generated, whether there is a decarboxylation there. You don't need to mention that. Just tell me it converts this to this, because it mentions using, state the roles of the following enzymes. And then phenylalanine hydroxylase. That was another question which the students were finding a lot of challenges. Phenylalanine hydroxylase works much at the point we are breaking down phenylalanine to tyrosine. Hydroxylase. If you look at the structure of phenylalanine, there is no OH after the aromatic ring, the benzene ring there. So the addition of OH down there, that is the work of phenylalanine hydroxylase. And that is what was leading to phenylketonuria. That is what I wanted. I wanted the point where you are telling me that phenylalanine cannot be converted to tyrosine because the phenylalanine hydroxylase is defective. So the phenylalanine is transaminated to phenylpyruvate, phenylactate. And that is, you see, if you see the pyruvate, the second carbon has a keto. The ketonuria, there is a lot of ketones in the urine as well as the plasma. So at that point, you have to define the role of the enzyme. In this case, I want you to mention it converts phenylalanine to tyrosine. Point number, is it number 5? Differentiate between ketogenic and glucogenic amino acid and give one example each. Glucogenic and ketogenic. Based on the degradation products, amino acids are classified as ketogenic and glucogenic. So glucogenic by definition, their degradation products are pyruvate and any intermediate of the TCA cycle. The ketogenic, the degradation products are the ketone bodies, which are beta hydroxybutyrate, acetoacetate, and acetone. The best examples of glucogenic, there are very many. The one which we've just seen, there's asparagine, aspartate, glutamine, glutamate, glycine, serine. There are many. Histidines, threonines. But then for the ketogenic, they're not that much. The phenylalanine, which you're seeing here. But the phenylalanine and tyrosine are both ketogenic and glucogenic. Only lysine and leucine are purely ketogenic. So the ketogenic ones, purely, they are lysine and leucine, because their degradation products will only give specifically the acetoacetate. So just by mentioning, just define the degradation product of glucogenic amino acids are pyruvate and any of those intermediates of the TCA cycle. You know all the intermediates. It can be fumarate, it can be succinyl-CoA, it can be alpha-KG, it can be oxaloacetate. If you see any of those together with pyruvate, it is glucogenic. But then for the ketogenic ones, purely ketogenic, if you see any of those, acetoacetate, acetone, or beta-hydroxybutyrate, then it is ketogenic. So just by defining those, you get 2 marks, and then the other 2 marks is for mentioning any 2 examples. So for each of them, the examples, I give half-half. So that's how I'll award. Just go straight to the point and answer. List the enzymes bypassed in gluconeogenesis and match them with their counterparts in glycolysis. List the enzymes bypassed in gluconeogenesis and match them with their counterparts in glycolysis. You know, the glycolysis will only take place when glucose is high. And I mentioned at the point glycolysis takes place, glycogenesis, synthesis of glycogen also takes place, because the excess of the glucose will be reserved in form of glycogen. Then, at that point, insulin as a hormone will regulate that process. Gluconeogenesis comes about when the glucose level is low. These 2 hormones, glucagon and insulin, work antagonistically. So when the glucose level is low, then it has to look for sources of generating glucose. So we talk about the non-carbohydrate precursors, which are glycerols, amino acid together with lactate. So if you talk about the bypassed steps, and that is why we always stress much on the irreversible steps when it comes to glycolytic pathway. Because during gluconeogenesis, as much as the arrow will go towards synthesis of glucose, because the body is under starvation, so it is looking for sources of glucose. Because you know the brain needs glucose, but that glucose is low. So we need to find sources of where we need to get glucose. So it has to come from those amino acids, which will connect via the pyruvate, which will connect via the oxaloacetate, and then go straight to formation of glucose. So the arrow for gluconeogenesis goes towards glucose, while the arrow for glycolysis goes towards pyruvate. So in that case, if you talk about the bypass steps, the irreversible steps in glycolysis are the bypass steps in gluconeogenesis. So if you talk about their counterparts, in glycolysis, we find the hexokinase as step 1, the phosphofructokinase as step 3, and step 10, we have the pyruvate kinase. But now, in gluconeogenesis, the first step will have the 2 enzymes, the pyruvate carboxylase and phosphoenol pyruvate carboxykinase, catalyzing those steps. So, and then you have the second bypass, where you have the fructose 1,6-bisphosphatase. Then lastly, you have the third bypass, where you have glucose 6-phosphatase, catalyzing the formation of glucose from glucose 6-phosphate. So just by matching, because at the end where you have the hexokinase, you'll have now glucose 6-phosphatase. At the end where you have the PFK in glycolysis, you will have the fructose 1,6-bisphosphatase. At the end now where you have the pyruvate kinase, It will be the first bypass in gluconeogenesis, where you are going to have the phosphoenol pyruvate carboxykinase and the pyruvate carboxylase. So just to be precise with our answers. So the next point says, using structures, show the steps involved in stage 2 of glycolysis. I already mentioned the glycolysis have 3 stages. Stage 1, destabilizing our glucose. The D-glucose is destabilized until we get fructose 1,6-bisphosphate. So by destabilizing our glucose, we have the 3 steps in stage 1. And that stage 1 is where the ATP utilization steps take place. But now you have stage 2, where you have step 4 and step 5, the aldol cleavage taking place, the isomerization taking place. So there is interconversion of an aldose to a ketose to an aldose. So that is the stage 2, where we are going to cleave the 6 carbon, fructose 1,6-bisphosphate, to 2 of the trioses. Another question says, using structures, show the 1-step degradation of alanine. Alanine is broken down to pyruvate, glucogenic amino acids. The alpha Kg and glutamate, they are carriers of the amino group, which comes from the amino terminal end of that alanine to form pyruvate. The enzyme is always an alanine aminotransferase, an important enzyme if you want to know the liver damage. Aspartate and alanine aminotransferases, very important. So that is the enzyme, and it is, that is the easiest one, because even at the time I try to set the questions, I'm trying to be lenient, because the student shall already know the simplest structure of an amino acid, alanine. And it's only 1 step. Already pyruvate, you've come across it in glycolysis. I told you, for you to understand metabolism, you just have to know glycolytic pathway and gluconeogenesis, to be sure. So it finds when the students already know the structure of pyruvate. Just that 1 step conversion. And then you have your 4 marks. So you have to write the correct enzyme. So that is glucogenic amino acid. Degradation of alanine will always give us pyruvate. And that is the definition of glucogenic amino acid. Another question says, where does beta oxidation take place? I think that one you already did. And the 4 steps involved, you already did that. Short notes on phenylketonuria. Short notes on phenylketonuria. I said, phenylketonuria, one of the metabolic disorders. I already uploaded the amino acid degradation, their classification as glucogenic and ketogenic, and both glucogenic and ketogenic, and I also uploaded urea cycle. By tomorrow, I shall upload something on lipid metabolism so that you can revise and try to get something and see on how you can answer the questions. But now it says that write short notes on phenylketonuria. It is best match on the degradation of phenylalanine to tyrosine. So the enzyme phenylalanine hydroxylase is defective. Phenylalanine cannot be converted to tyrosine. And that enzyme is the one which controls the whole pathway, because that is the same pathway which involves the generation of the catecholamines. The norepinephrines and the epinephrines are generated there. The dopachinone, which leads to formation of melanin, is there. So albinism will also be as a result of that. So that first step, conversion to tyrosine, very important. So if that enzyme is defective, phenylalanine is not converted to tyrosine. There is transamination of phenylalanine to phenylpyruvate, phenylactate, and there is also high concentration or elevated levels of phenylalanine. So you'll find the ketones, because it's forming the phenylpyruvate and the phenylactates. You'll find the ketones, high levels of ketones in the urine as well as in the blood plasma. So in such a case, you talk about a phenylketonuria, the name keto. That is how important it is. As much as you're going to mention for me some of the characteristics or the features of the patients who might be found with such a disorder, but it's very important to mention the type of enzyme which is defective, what is not converted to what, and that is what leads to high levels of phenylalanine. But now it is transaminated to phenylpyruvate and phenylactate. And that's why you can test the presence of ketones in the urine as well as in the plasma. Another one, write short notes on alkaptonuria. Still the same pathway. Defective enzyme, homogentisic acid oxidase. Very important. And that's why we mentioned in alkaptonuria, if you find the diapers of the kids who have such a condition, when it's exposed to air for some time, it will turn black. But even the urine of somebody with alkaptonuria, it will be black. But then the normal urine will have its normal color. But then this one, which has a patient who is suffering from such, will have the urine dark. So when it's exposed to air, it turns black. And still, if you do test the intervertebral discs, there will also be some black deposits. So you have to mention the enzyme, because we are talking about a metabolic pathway. What is not converted to what? And what is high? The high levels of what? Which leads to such a condition. So it's very important to mention such before we can go straight and write on the characteristics or symptoms related to such. But remember, phenylketonuria is also, it is an autosomal recessive disorder. We'll understand much when we go to genetics. But then, for the autosomal recessive disorders, they're only expressed in homozygous recessive state. So if it's in the case of a carrier, it's not expressed, because the dominant one for healthiness is going to suppress the one allele which carries for the disease. So it won't be able to show. But in the case where it is homozygous recessive, it's going to show. So at the point of genetics, we'll be able to understand how phenylketonuria comes about. But at this point, we have to explain much in relation to metabolism to understand that defect. There is a gene which codes for that enzyme you're seeing there. The gene product is that protein. So that's why you can see some of them, they have to be interrelated, because you talk about the central dogma if you'll be doing some molecular biology. But let me not mention much for now. So the alkaptonuria, the phenylketonuria is the same pathway. It relates much, the degradation of phenylalanine to tyrosine. And then further, it will go to the formation of the ketogenic as well as the glucogenic degradation products, because they fall under both ketogenic and glucogenic. This one still talks much about defined gluconeogenesis, of which I don't bring definition nowadays. I don't bring. And name key enzymes of gluconeogenesis. No, I don't bring that one. Write short notes on how ketosis result. So it relates much with the lipid metabolism. We are talking much about a situation where fatty acid oxidation has taken place. Acetyl-CoAs have been generated. Then the acetyl-CoA has to go to the TCA cycle for formation of the reducing equivalences, which are used as electron carriers. But when they go to the TCA cycle, they find oxaloacetate insufficient. So it will lead to formation of ketone bodies. The acetoacetate will be forming in high levels. And then you'll be able to form the acetone and the beta hydroxybutyrate as well. So the ketone bodies will be able to form because the acetyl-CoA has to find the oxaloacetate available in the TCA cycle to form the products in this TCA cycle so that we can generate the reducing equivalences. But when it finds oxaloacetate insufficient, then it will resort to formation of ketone bodies. That is how the level of the ketones will be high. So it will be used in ketone bodies formation instead of being used in the TCA cycle for generation of reducing equivalences FADH2 and NADH, which are electron carriers in electron transport chain. Still, I'm at section C. I've touched a bit of them. I've touched a bit of the carbohydrate metabolism. Not necessarily should we discuss everything, but very important to touch several questions in each of those metabolic pathways, carbohydrate, lipid, though I didn't represent the nucleic acids or the nucleotide degradation, the purines and the pyrimidines, but we touch each of those so that you can have an idea which type of questions can be asked. You can't also set questions based on one metabolic pathway, maybe carbohydrate only. No. You have to mix them with protein metabolism, lipid metabolism, and if I didn't even touch urea cycle, which I might touch, it doesn't mean that it won't come. I don't say that whatever I'm discussing is what is going to come. I'm always trying to tell you much on what to expect and how to prepare. Now that you just have a day to go, but there is still time. So you need to reorganize yourself and read well and straight to the point when you're asked. Using structures. For me, structures and the correct enzymes matter much. Don't write stories. Only if you're told to write short notes. There are some, like alkaptonuria. I've not said using structures. Like phenylketonuria, like ketosis. Maybe 4 marks. Just give short notes on how it comes about. So, in the essay is where you give a broader picture. Even if you are involving a metabolic pathway, like the first one says, using structures outline the tricarboxylic acid cycle and its biological importance. So when you talk about outline, clearly show the structures of the acetyl-CoA, how it comes in to combine with oxaloacetate to form citrate, how the citrate, the isocitrate, the enzyme involved there, isocitrate, the alpha-KG, how it goes, succinyl-CoA to succinate, to fumarate, to malate. You have to show all those 8 steps and whatever is generated, what goes in and what goes out, those structures. Because in the essay, that is where you need to discuss all which is needed. So don't discuss it like an essay, You just talk about whatever happens on the enzyme. I need structures. I said, where I can exclude structures is for nutrition students. As much as they're supposed to know, but for the nursing, the medicine, pharmacy, we should understand structures. At a certain point, a reaction, what happens? The product which is forming, what type of process was going on there? What type of reaction was going on there? And this enzyme, we are told about the core enzyme, the core factors. What is needed there? How important are the vitamins? How important are the minerals? So you have to explain using structures and the correct enzymes. So in this case, I need the tricarboxylic and in a cyclic form. In the metabolic pathways types, the glycolytic pathway is a linear. So there is no way you can draw glycolytic pathway in a cyclic form. It is linear. You have the substrate, the enzyme, the substrate binds the enzyme active site, you have the formation of a product. Then the next step, the product is now the substrate. That's how it goes. But the cyclic, the same enzymes, it's a cycle. Availability of acetyl-CoA, oxaloacetate, forms the citrate. Availability of carbamoyl phosphate, the way it joins to form citrulline, ornithine has to combine with the carbamoyl phosphate. So the urea cycle, cyclic, TCA cycle, cyclic. And then you come to fatty acid oxidation. It is spiral. So just by describing the TCA cycle in a cyclic manner, just give me the answer, the structures, the correct enzymes, and then the biological importance, the generation of the reducing equivalences, how important the FADH2 and the NADH will be important as electron carriers in the electron transport chain. There also ATP is going to be generated, the oxidative phosphorylation. Then point number 2 says, without structures. Why did I say this one, without structures? No, I've read it wrongly. Using structures, discuss the oxidative and the non-oxidative phase of the pentose phosphate pathway and name the enzyme regulating the pathway. You know the oxidative phase. First, the main objective of the pentose phosphate pathway is first, it has to generate the NADPH, which is used in fatty acid synthesis. The NADPH for the synthesis of the steroid hormones, the estrogens, the testosterones, they need NADPH. Integrity of red blood cells, they need NADPH. Mostly in lipid metabolism, NADPH is going to be needed. So it comes from the pentose phosphate pathway. So the pentose phosphate pathway is going to generate NADPH. It is going to generate ribose. We said in our introduction in the sessions, in the channel, the nucleic acids, the building blocks are the nucleotides. If you talk about the nucleic acids are the DNA and the RNA. If you look at the building blocks, the nucleotide, it has a phosphate, it has a base, and it has a pentose sugar. We talked about fructose and the pentoses forming a furanose, the 5-membered ring. That ribose sugar is generated by the pentose phosphate pathway. So after forming the ribose 5, the work of phosphatases, you know phosphatases work opposite of kinase. By adding water, they are going to remove the phosphate at carbon atom number 5. You remain with ribose. So the DNA and the RNAs will have, the RNA will have the ribonucleic, the deoxyribo will be for the DNA. So it generates the ribose for the nucleic acids. And thirdly, based on different lecturers, but in my case, I say ATP. It acts as a shunt. Remember in the non-oxidative phase, we generate glyceraldehyde 3-phosphate, which can go to the glycolytic pathway in stage 3 to generate energy in form of ATP. It generates fructose 6-phosphate, intermediate of the glycolytic pathway. So it acts as a shunt. It can also be an alternative way to generate energy in form of ATP. So those are the 3 which are involved in the pentose phosphate pathway, the products which are released there. So it says, discuss the oxidative phase. The NADPH is only produced in the oxidative phase. I said, depending on the need of the cell. The cell can be in need of NADPH for all those pathways where lipid metabolism is involved, fatty acid synthesis, all those, whatever, synthesis of the steroid hormones. So only the oxidative phase generates NADPH, and it's only generated in step 1 and step 3. The enzyme controlling all this is glucose 6-phosphate dehydrogenase, the first step. So the oxidative phase will generate NADPH, which is used by these pathways, which needs it. And in the non-oxidative phase, We talked about the interconversion of sugars. So you'll find the ketolases, the transaldolases, but there are also epimerases and isomerases. Epimers do not only occur on galactose and glucose or mannose and glucose, even the ribulose and xylulose will be epimers. So based on the reaction, the International Union of Biochemistry thought of classifying those enzymes based on the reactions. So the epimerases will be there, isomerases will be there, conversion of ribulose 5-phosphate to ribose 5-phosphate. Because one is a ketopentose, which is ribulose 5, and ribose 5 is an aldopentose. The interconversion, that is the non-oxidative phase. So it is just a matter of balancing. You have 5 carbons plus 5 carbons, you have 10. Then you'll form the 7 carbons, the pseudoheptulose and glyceraldehyde 3-phosphate. The next step, you'll have the pseudoheptulose and the glyceraldehyde. It means it is from a keto donor to an aldose acceptor. But based on the number of carbon atoms which are transferred from a keto donor to an aldose acceptor, the 7 plus 3 are going to form 6 and 4. Erythrose and fructose 6-phosphate. So that's why I'm saying in the non-oxidative phase, we can generate the ribose and the ATP because the glyceraldehyde 3-phosphate and the fructose 6-phosphate will go to the glycolytic pathway. So the second point says, outline the urea cycle and any of 2 diseases related to the pathway or associated with the pathway, the defective enzymes. At a certain point, if agenosuccinate lyase is defective, what happens? If agenosuccinate synthase is defective, what happens? Remember at that point, citrulline have to combine with aspartate. What happens? And imagine the importance of urea cycle. If those amino acids degradation, the ammonia produced, the nitrogen component produced by degradation of those amino acids and digestion of proteins is left in the system, it's toxic, which can at last lead to death. So the urea cycle make it possible for the terrestrial animals, just like us, human beings, leave alone the aquatic ones, which comes as ammonia, the birds, which come as uric acid, but we are talking about ourselves. So it will not reach that step 4, where urea is released. So any of those enzymes which are defective in that pathway, you have to describe a disease related to that, but you have to describe the pathway as well. You start very well by formation of carbamoyl phosphate. The presence of that ammonia, the NH3, which comes from the degradation of those amino acid and digestion of those proteins, you have to show it well. The carbonic acid and ATP as a source of energy, it's going to give the carbamoyl phosphate. So the phosphate will come from the ATP which you're seeing there. The carbamoyl phosphate synthetase getting involved, and then the carbamoyl phosphate has to combine with ornithine to form citrulline. So describe, using structures, the correct enzymes, and then mention any of the 2 diseases which are related to any of the defective enzymes in that pathway. So just being precise, discuss reaction sequence of the urea cycle and note the disorders. So that is the same. Discuss fatty acid synthesis using structures. You know, synthesis takes place in the cytosol. I just mentioned how important the organelles are. But we started by looking at the biomolecules, the components in those organelles. The reaction which takes place in those organelles is what we are discussing now. And then it relates with a disease associated with any of the defective enzymes. That is now you are coming to clinical biochemistry, because you have to test a certain chemical component when there is a disease associated with such. In the case of phenylketonuria, a chemical component which you're looking at there is the ketone. It is more than normal, but then it is present in urine and blood plasma. So you have to get the correct sample. Not that if you want urine sampled, then you get from the patient maybe mucus. You won't get what you want. So you have to get the correct sample, get the correct quantity, do the correct test. That one goes much with biochemistry 3, the clinical biochemistry. After covering biomolecules, you cover metabolic pathways, so you understand much the organ function tests based on carbohydrate metabolism, based on heme metabolism. Somebody talks about jaundice. Where did you see bilirubin? You talk about the unconjugated and conjugated bilirubin in relation to the type of jaundice. Maybe you are talking about obstructive jaundice, hemolytic jaundice, hepatic jaundice. What is the chemical component which you are trying to test there? And the metabolic pathway involved. It is heme metabolism. Which one? Degradation or synthesis? You talk about porphyrias. Degradation of heme or synthesis of heme? So if you are talking about the degradation of heme, definitely you have to talk about the jaundice, the bilirubin will be related to that. But synthesis of heme, the porphyrias. So at that point where the defective enzyme, there is an enzyme which is defective. How are you going to relate the chemical component which you are testing with the type of metabolic pathway? What should we be testing? So in this case, we are talking about disorders in relation to urea. This case, we are talking about the fatty acid synthesis. So the fatty acid synthesis takes place in the cytosol. The degradation or beta oxidation takes place in the mitochondria and in the inner mitochondrial membrane. Remember the outer, the carnitine has to transfer it. Is it transferring or ferry it to the inner mitochondrial membrane? And even in fatty acid synthesis, remember, the first point here is it has to be ferried because degradation takes place in the mitochondria. You have to get a citrate shuttle. It has to shuttle our acetyl-CoA into the cytosol from the mitochondria. Then, the acetyl-CoA itself, there has to be carboxylation to form malonyl. After the malonyl now, they'll condense. But now, acyl carrier protein is involved. And in the case where we had the oxidation in degradation, in this case, we have reduction. In the case where we had hydration, in this case, we have dehydration. So it goes on and on as you lengthen the fatty acid length. So you have the enzymes involved. So it involves also the steps, the condensation, the reduction, the dehydration, and then you'll be able to build your saturated fatty acid. So you have to use the structures. But remember, in the fatty acid synthesis, it needs NADPH. Those steps which involves reduction, NADPH is used, and this NADPH comes from the pentose phosphate pathway. I talked about interactions of these pathways. Imagine the NADPH from the pentose phosphate pathway, carbohydrate metabolism. Now it has to go to the synthesis of the fatty acid, lipid metabolism. And it still connects with the nucleic acid, the purine and the pyrimidines. So that is how important the system works. That is the sweetness of biochemistry. We have to understand how the reaction takes place. But before we understand how the reaction takes place, we have to understand first the biomolecules, the components, how important the organelles are. The cell can't exist without the organelles. And each of those organelles have a specific function. Remember the cell division, cell differentiation, the mitochondria, the cytosols, the nucleus, carrying almost 95% of the DNA, and the DNA is the hereditary material. Look at how interesting it is. So we have to understand the process, and that is the work of the metabolism. So in fatty acid synthesis, explain well whatever happens there. It is opposite of whatever happens in the oxidation. Using structures, outline gluconeogenic pathway and its medical importance. Gluconeogenesis, that is the simplest question. Because the arrow, I always look at the arrow, even using structures, the arrow should go towards, and you should understand at what point is this metabolic pathway important. During starvation, what happens? It has to look for sources because glucose is needed. The brain needs glucose, but glucose from glycogen is not enough. It just survives for only 24 hours. So it needs to get glucose from other sources, like amino acid, like glycerol. So the amino acids, aspartates, the good example, it will go via the oxaloacetate. Some like glycine, which will definitely convert to pyruvate or alanine. That's how important it is. So the arrow should go. Imagine a student writing very correct structures and correct enzymes. But since the arrow is going towards formation of pyruvate, I don't give a mark. I write a 0. Because first, you didn't understand the definition of gluconeogenesis. Synthesis of glucose, arrow should go towards glucose. And all the reversible steps, the enzymes remain the same. In fact, the only structure which you are introducing here is oxaloacetate, and it finds when the oxaloacetate is the simplest structure so far. You've already seen it in the TCA cycle. So it is very important to understand the question where the arrow goes. The bypassed steps, they are now having new enzyme. The bypassed steps. Bypass 1, specifically state bypass 1. Pyruvate to oxaloacetate, pyruvate carboxylase. Bypass 2, fructose 1,6 bisphosphate to fructose 6-phosphate, bisphosphatase. Bypass 3, now we are forming our glucose. Glucose 6-phosphate to glucose. Very simple, and its medical importance. Remember how important the glucose is. So we need to find the sources of glucose from the non-carbohydrate precursors, and that one will need a little bit of explanation in words after drawing the structures. Another question says, using structures, outline only the bypassed steps. For the makeup cut which I gave, I mentioned using structures, discuss gluconeogenic pathway or gluconeogenesis. And somebody still just give me the bypassed, and it is 20 marks for heaven's sake. If you draw them correctly, it's okay. Since it's a cut, I'll give you a mark where you drew the correct and wrote the correct enzyme. But then the question says the whole pathway. And then this one, if you tell them only the bypass, now they give you the whole pathway. If you look at the marks which is in bracket, know the limit of answering the question. For the bypassed, I'll only give 10 marks. What I need is state very well bypass 1. Pyruvate, oxaloacetate. Oxaloacetate, PP. Go straight bypass 2. And some people are still writing kinase. For heaven's sake. That is not a kinase. Removal of a phosphate at carbon atom number 1. Phosphatase is doing the work, please. Add water. Don't put ATP, because now the whole thing is wrong. Let us just be precise with our answers so that we don't make small mistakes which will cost us. If it is a phosphatase, there is nowhere ATP is coming in. ATP comes in the case of kinases. So if you see a phosphatase, it is a class of hydrolases. Water will come in to remove that phosphate. So bypass 2 will have the fructose 1,6 bisphosphatase to fructose 6-phosphate. Then lastly, the bypass 3 will have glucose 6 phosphatase to glucose. Then we have diseases. No, there is one saying, please, sorry for that. Using structures, discuss the 3 steps catalyzed by the core enzyme in the pathway involving entry of galactose to glycolysis. The 3 core enzymes in galactose metabolism. We are talking about galactokinase, step number 1, which is involved in phosphorylation. We are talking about uridyl transferase activation of galactose by adding uracil or uridine, which comes as UDP, glucose. Then we are talking about the last one, which is very important, epimerase. It has to convert our galactose to glucose because there are epimers at carbon atom number 4. Those are the core ones. So give me the structures for that step, where galactokinase catalyzes, where uridyl transferase catalyzes, and lastly, where epimerase catalyzes. That's how simple it is. It is 10 marks. And then, using structures, outline degradation of any 3 glucogenic amino acids. That is 10 marks. So, still, we had a lot. Glucogenic amino acids, I've mentioned their degradation products are pyruvate and any intermediate of the TCA cycle. So if you have asparagine and aspartate, those are already 2. I mentioned it. It's going to give us oxaloacetate, give the structures very well and the correct enzyme. If you have glutamine and glutamate giving alpha Kg, those are already 4. Then you have the simple step of alanine to pyruvate. You can't miss to discuss using structures. So just any 3. Whether you take alanine, asparagine and aspartate, or alanine, glutamate and glutamine, whichever is easier for you, which you can discuss. And then the other question says, discuss any 2 diseases associated with degradation of aromatic amino acids. That is 10 marks. So discuss about alkaptonuria, discuss about phenylketonuria. You can discuss about albinism. The most point here which you need to stress much is the defective enzyme, what is converted to what is high, what is elevated, and then now is when you can give the characteristics related to such a condition, and then definitely treatment. But it finds when you've already discussed the defective enzyme and what is supposed to be converted to, but at this point it's not converted to that product, then that leads to such a condition. Explain how ketosis results. I already mentioned that at the time the acetyl-CoA is available from the degradation of fatty acid, from conversion of pyruvate in aerobic state to acetyl-CoA, the acetyl-CoA should enter the TCA cycle and find oxaloacetate available. But if it finds oxaloacetate not there or insufficient, then it will lead to elevated levels of the ketone bodies, and that is how the ketone bodies are going to be formed. Using structures, outline the pathway for ketone bodies formation. It starts with 2 acetyl-CoAs. You form acetoacetyl, you know how the story goes. until you form. The first ketone body to be formed is acetoacetate. Then from acetoacetate, one step, dehydrogenase getting involved, one step, dehydrogenase getting involved. You are going to form acetone and beta hydroxybutyrate. Describe them well. Don't confuse them with beta oxidation. And with beta oxidation, it was very easy. You start with an activated fatty acid CoA. Then the next step, formation of a double bond. You know, water has to be added to remove the double bond. There is formation of an hydroxyl. Then after that, imagine formation of a keto. Then lysis. So thiolysis will take place and you'll have your fatty acid, which you started with, 2 carbon less because acetyl-CoA is generated. So I said for palmitate, you will generate 8 of the acetyl-CoA. For the stearic acid, you will generate 9 of the acetyl-CoAs. I think people are going down and down, but these ones are looking the same. 5 glucogenic bypassed steps because it's already late, it's 11. Substrate level phosphorylation steps in glycolysis using structures, 8 marks. You know the substrate level phosphorylation steps, they are in stage 3. The ATP generation steps. Step number 7, phosphoglycerate kinase, and the last one is pyruvate kinase. So only those 2 steps, they are in stage 3, where ATP is generated. Discuss. Using structures, discuss reaction sequence for beta oxidation of fatty acid. That is what I've just discussed. Formation of the double bond, formation of hydroxyl, formation of a keto, and then reaction for ketone bodies formation. So I was just discussing a few facts, how you need to answer them, and now I'll call it a night. Thank you for everyone. I can't see the comments, so I'm sorry because my phone, I can't see. But I think Vutu, you are still there. Just have a lovely good night. And I wish you the best as you prepare for the exams. I'm going to upload some of the videos so that you can revise. It can help you in revision and pick what you didn't get. Just by following the video, I hope it will be helpful to the students. Thank you all for the subscription. Thank you all for your time, for the likes, for the comments. I know we'll be able to move a step ahead, and I hope it will be helpful to the students. So thank you so much, and God bless.